Okay. So we are delighted to have today with us in our Bar-Ilan University Vision Science Seminar, Professor Eli Brenner from the, I think I'm Vrij, University of Amsterdam. Eli started as a biochemist and moved to physiology and neuroscience. He did his BSc in biochemistry at the University of Utrecht and then a PhD at the Brain Research Institute at the University of Amsterdam, which is today known as the Netherlands Institute of Neuroscience. And he did this under the supervision of Mirmiran about neuropharmacology. Eli Brenner is a professor at the Faculty of Human Movement Sciences at his university in Amsterdam. His research focuses on how vision is used to guide human actions, as well as additional many aspects of perception and action. His investigations look at vision from a naturalistic in the wild perspective on top of lab-based investigations, taking into account the fact that humans are dynamic entities moving in and interacting with their environments, and his innovative research actively goes into the wild to investigate our vision. He examines both perception, eye movement, different properties of vision such as motion, color, proprioception, distance, illusions, interactions with other senses, and much more. Professor Brenner has authored hundreds of scientific papers and has an H-index of 57 according to Google Scholar. So without further ado, I'd like to welcome you, Eli, to our Vision Science Seminar to hear about your exciting research, and thank you so much for joining us.
Oh, thank you for having me here, and I hope you didn't all join to hear stuff in the wild, because I'm going to talk about very in the lab experiments. So I'm going to just start my presentation. Oh no, I need to first share. No. Okay, I hope you can see my screen. If there's anything wrong, I don't see anyone now. I'm just talking at my own slides. So if there are any questions or if anything is unclear, just shout. So I'm going to talk about continuous guidance of human goal-directed movements. And I'm going to first, most of my time, I'm going to show you that people do this continuous guidance. And then at the end, I'm going to try to convince you that it's quite elaborate guidance. But I'll start by explaining why guidance might be very good. And I'm going to do this with a sort of a very simple simulation. So if you would have no guidance and you would make an error, and I'm going to just, as an example, use an aiming error. So the participants, so imagine you want to move from the black dot on the left to the one on the right, and your finger is that red dot moving dot. And you judge the direction wrong. So rather than moving along the dotted line, let me see, get it? Yeah, rather than moving along this dotted, do you see my cursor?
Yes, yes.
Yes, it is small, but it's visible in this case.
Okay, good. So rather than moving along the dotted line, you move along the solid line, and you'll end up somewhere up here. And so if you have this systematic error and you also have some variable error, the endpoints will look like these accumulating red dots here. So you'll have a lot of variability and be in the wrong place. Now, if you wouldn't just make this decision at the beginning and follow that, but you would continuously do this, so you wouldn't correct errors, but just continuously guide the hand with the error, you would get something like you see here on the right. So you would start off, of course, in the same way, but at any time, you will be reconsidering the direction which you go. So if we take a time here, the beginning of the gray line, at that moment, that you have already gone a bit wrong. So the direction to the target, the dashed line here, will be down here. So the error, this is the same error as you had in the beginning, but you'll now be moving along a shallower path. And of course, I say you'll now be moving. You won't be moving along that path because there is a delay in your system. So if you have seen it here and decide, oh, this is the direction I should be moving in, you will only actually move in that direction a bit later. And of course, that will mean that you move, you still curve, but you're going to gradually move down. And as it will be obvious from this, as you get closer, you notice the direction more, so that the error you'll curve more, and you'll end up with points as shown down here. So just by doing the same thing all the time, you'll move from having this sort of error to this sort of error. Without correcting the error, you're still making the same systematic and variable error in your movements. So this is why it might be advantageous to continuously online guide your hand movement, foot movement, whatever. And so most of my talk, I'm going to show you that people actually do this. And I know that some people in the audience have already seen my presentations about this, but too bad. So what we did is we developed a method to try to demonstrate continuous guidance. There have been many examples of showing that if something happens, you can respond to it, but we want to show that you're guiding it all the time. And of course, we can't really show all the time. So what I'm going to show is at 120 hertz at least. So what we did is we had people do a really stupid task, which is to start with your finger at a certain position, and then just a target appears, and the participant has to lift their finger and tap on the target. And the target is jittering. And then they tap on it and it stops. So I think you see the jitter here, move your finger and the jitter. Very boring for the participant. They do this lots and lots of times. The interesting stuff all comes in the analysis. So I'm not going to talk anymore about the experiment now. I'm going to talk about the analysis. So if you look at a trial, this jitter isn't just any jitter. It's a random walk, meaning that at every moment, the target is as likely to go to the left as to go to the right. And this happens at 120 hertz. And the steps are 1.7 millimeters. So they're very small steps. And the target is much wider than, I think it was 2 centimeters diameter. So it's really little steps in it, but you can have consecutive steps in the same direction or not. So here I've plotted just a part of one trial. So I've plotted the lateral position of the target as a function of time or consecutive steps. And so in this interval, it moved right, 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 then left, left, right, 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 left, 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 right, left, right, left, right, right. This is just one trial, but of course, it's completely random. So if we take another trial, it'll look different. And if we have a lot of trials, you'll have a lot of different steps in all sorts of directions. And the trick that we do in the analysis is to first align all the trials in some way. You can do it at the beginning for reasons that I can explain later if you're interested. We align them at the end, so at the moment of the tap. And so that's why in this example, but of course it's a schematic example, they don't all start at the same time. And you see just jumps going all over the place, and they have two colors, and you may have noticed that the colors are not completely random. So I take all the trials and I select a certain moment. In this case, I'm showing the 7th step from the end. You take all the trials and split them by whether that step was to the left or to the right. So in this case, all the ones which stepped to the right upwards are shown in blue, and all the ones that step to the left are shown in purple. So during the selected step, don't see my cursor anymore myself. So now if I take from those two groups of trials, I've split all my trials into two, I take the target motion, then you will see a pattern like this. So if you took at the mean lateral position of the target on all the trials in each of the two groups, then they will both be the same, just at the starting position until the step that I selected, because it's randomly left and right, completely independent of each other. And then on that one step, you have a step either to the left or to the right. And then after that, it's again the same. So it will look as if I've just done an experiment with one little step. If for these small steps, the responses are more or less proportional to the steps. Of course, that's an assumption I'm making here, but as you'll see, it's true. So this is what the target does. And if I now take these same two sets of trials and I look at what the hand does, and I'm going to now only show the lateral position of the hand, or of the finger actually, because I'm recording the finger position, then you see a response. So here I'm plotting the response, which is the difference in the lateral finger velocity as a function of time from the selected step. And you can see that after 100 milliseconds, something happens. There's a peak upwards, which is my response. And so this might sound very mysterious, but I'm going to take you through the steps of finding this response. This is actually data of a different study, but the response is always measured in the same way. And I have nice pictures of this study. So this is actually a study in which the background moved. It wasn't the target that jumped, but you get a similar sort of response. So here on the left, you see 20 trials of the finger moving. It's more or less the same task. There's a target appears at the top, and there's a starting position at the bottom. And participants, as soon as the target appeared, they have to just move their hand up and tap on it. And so you can see a lot of paths going upwards. And you can also see that there are two colors. And in this case, the colors represent the direction in which the background moves. So it moved either to the left or to the right, shown in red or blue. And you could just say, okay, I can see here at the top that something has happened. They're sort of split a bit. But we want to actually measure the timing of the response and things like that. And there's a problem there, which is that the dots, you've probably noticed there's a lot of dots on these paths. The dots represent the same time on different trials. So there isn't only a difference in how people move left, right, but there's also a difference in how quickly they move. No, I can't. Oh, sorry, I can't find my cursor, but does anyone see my cursor?
I can't.
No, it's disappeared.
No, we don't see.
It doesn't matter. I don't need it. I have a strange bar which I can't get rid of on the screen.
You want to try to share again?
No, it's fine. It's fine.
Okay, sorry.
So there, you can see that there are also different times at which. So they're not synchronized in time, and they're not synchronized in any way. So now we're going to synchronize them as a function of some event. In this case, it's the background starting to move. In the actual experiment that I've been talking about, it's the selected step. So you take the time from then, and then just plot the lateral position. So the positions that you see on these trajectories on the left, they're now just flipped around so that up is right and down is left, and in time is to the right in the panel on the right. And so you see a lot of wiggles. It looks even bigger because I've extended the scale, of course. But you can see if you look at the average paths, so I've now drawn in thick lines the averages of these paths, you can see that something is happening at about 100 milliseconds or a bit after 100 milliseconds. Suddenly the red and the blue curves are not doing the same thing, but the blue one is going up and the red one is going down. And you can see this, of course, then even clearer if you don't plot the lateral position, which I've just repeated that figure on the left here, but you plot the lateral velocity, which is the figure on the right here. So that's just the slopes of the left figure are shown in the right figure. And then you can see that the red and the blue are doing the same sort of thing until about 100 milliseconds, and then they differ. And of course, what we're interested in is not whether the hand curves a bit left-right, as you see on the left side of the right panel, but the difference between the response to the event, so the difference between the red and the blue curves. And so you could also just plot that difference. And so that's what I do here. So again, the left panel is just the same thing. Again, the right panel shows the difference between the red and the blue curves. And now actually the thick purple curve is the difference between those two, but you can see that it looks the same, but it's actually not the difference between just these 10 and 10 trials, but of all the trials in that of that participant. And the other ones show data for other participants. So you can see that all participants have some sort of a response. It's similar. That's not exactly the same for all participants. And there can be many reasons for that, which we can also discuss later. But I'm going to show you all the data I'm going to show you are averaged across participants after this. So I'm going back to the figure of the response to the selected step. And so now you should hopefully understand what this figure is showing you. If not, stop me now and I'll explain it again. But now, of course, you probably realize I've selected a step, but I can select any step I like, right? This is just one step. And so I can do the same for a lot of different steps. And so these are responses for a lot of different steps with the color coding shown at the top. So if you can see a clear order in these things, which is that if you select a step long before the tap, and long before in this case is 300 milliseconds, you have quite a small response. So that's the lowest red curve. And well, the 100 milliseconds actually stops, of course, then after 100 milliseconds, but you can see some of the blue curves starting to go up and then suddenly stopping because at that time you already hit the screen. And then, so the more time you have, the slower, the less vigorous the response. And this makes sense because, in fact, what you're doing is you're just trying to smoothly correct for the whole error that you see, this little bit of jitter, which for your movement to hit the target can be interpreted as an error. You're trying to correct that in the remaining time. And so the more time there is, the slower you can do your correction. So this is for a jittering target. And I think this shows that you are all the time correcting, and yet you're not just responding to the jitter, you're actually doing it in a meaningful manner to make sure you reach you do whatever adjustment you need to do in the time that you still have. In this case, you can see that the target is jittering. Although it's a small jitter, you do see that it's not stable. So we can also do the experiment slightly differently. And I've got an example here, which I hope looks okay on your screens. It will depend on your internet connection and mine. So I hope you see me tapping on targets. And in this case, the target is jittering, but of course you can't see the jitter now because I've just added a rightward motion to the target. So the target is moving to the right and at the same time jittering in the way that I just showed, but participants can't see that. So you can't see it when you're actually looking at it, so you certainly shouldn't be able to see it in this version of it. And this also reminds me to tell you that people got feedback So if you hit it, you hear a sound, and if you miss it, it goes away from where your finger misses it. So
Ellie, can I ask a question?
yeah.
So first of all, I want to ask, were the people right-handers, all of them? I mean, was
That is a very good question. I'm not sure about in this experiment because in some experiments they're all right-handers, and in some experiments we also allow left-handers, and then have the target moving in the other direction.
Hmm,
Uh, I think in the static one we probably didn't make any distinction, so I could guess that there are also left-handed people in it,
I see.
but
Okay.
mostly they're right-handed participants.
Okay, thank you.
You can do it with both hands, not
Oh, okay. Mm-hmm.
yeah. There's no the yeah. Uh, so this is this is just the same thing. I'm going to be very short about this. I just thought that seeing what it actually looks like might help. Uh, oh, so this is what the data then look like for the same. This is the same sort of experiment. So on the top right is the data I showed you before, and in the center is the data with when the target is moving. There are some small differences which we can discuss in the end if you want, but in the main effects are all the same. So you again see the adjustments irrespective of when the target jumped. So now this is about the target. So now I started thinking, well, is this just something that we do with a target? I know that in these tasks you're always looking at the target. The target is the critical thing. So what about your own movement? So we wanted to do the same thing for your own movement. So jitter your hand, but of course I can't actually jitter a person's hand. So instead of that, we use this sort of a mirror setup, which I don't know whether you're all familiar with this sort of setup, so I'm going to explain it anyway. So there are three surfaces. So this is the picture on the left. And the top surface is a projection screen, a back projection screen. The central surface has a mirror in it, and the lowest surface is the surface on which you move your hand. And the distance between the lower surface and the mirror is the same as between the mirror and the projection screen. So that means that when you look into the mirror, it looks as if the things on the projection screen coincide with the surface that you're moving your hand on below the mirror. And of course, this is all just to hide the hand as you move from the starting point to the target. And we're measuring, of course, the participants' finger movements again. And we now present this position as a cursor on the projection screen. So they actually see a cursor moving with their finger. And the whole point about this is that then, of course, I can jitter the cursor around. So the task here was slightly different. It wasn't to tap on the target, but to just sweep your finger through the target. And either the target jittered in the same way as in the experiment that I just showed you, or else the cursor jittered, and it jittered in the same way as in the first experiment I told you, so just static relative to the finger. But when the finger moved, it also moved along with the finger. So it was the jitter followed a random walk with respect to where the finger was. And then we do all the same tricks that I showed you before. This is from a different paper in which, unfortunately, I wasn't consistent with my color coding. So now red is late and blue is early, but I guess you can figure that out. So on the left, you see the dots of where the finger was at the different times. I now show also much earlier movements because this setup works at 60 hertz rather than 120 hertz. So I can draw more lines without it becoming a complete mess, although it still does look quite messy. And you can see, so in the panel called target steps, that's just a replication of what I showed just before. Only now it's a slightly different task. And you can see that you get a response. And for some reason in this experiment, it was a bit more than 100 milliseconds, which I still don't know why. And you can see that even when you take the green curves, which are actually I'm choosing a step well before the target even started moving, you still get the response. So this isn't surprising because it's just a sort of a replication, but it just shows that it also works in this task. And the panel on the right shows you the same thing for cursor steps. And you can see again that you see more or less the same thing. You probably notice that the effect seems to be smaller for the cursor. That shouldn't surprise us because for the target, you only have visual information. But for the cursor, yeah, you have the visual information of where the cursor is, but of course you feel it as being your hand, and you also feel your hand, and your hand isn't influenced by this jitter. So it's not surprising that this effect is smaller. So that's about continuous guidance, and I hope I've convinced you that you're actually continuously adjusting the way you move. So based on what you see, so that is continuous guidance. Now I'm going to try to convince you that actually this is not just a sort of a special thing for intercepting targets, but that you actually can consider all sorts of things when doing this. And I have all sorts of things, but I'm going to talk now about what I think is the most extreme, which is selecting a different target. Here I'm not going to use any more jitters, but I'm going to use the same sort of responses. And the task is slightly different. So if you have questions about the previous one and don't want me to start on something new, just ask.
Okay, so I do have one question about the fact that I mean it's a bit more than it took longer for the effect to show, and I wonder if, I mean, because it seems to me like it's a difference between closed loop and open loop. I mean, maybe the fact that you don't have consistent ongoing feedback from your own hand, maybe that delays your I don't know, I'm just
So there is a problem in the mirror setup, indeed, that you have a delay of about 50 milliseconds between your hand and the cursor. But of course, the response is to the cursor; it's not to your hand. So I don't see how that could make. But there is something in the setup that does this, because we've also done the slide through on the big screen. Where you do see your hand, and then you don't have the different delay. So I don't really know why. I don't know why this arises. I just pointed it out, but I have noticed that in different experiments you sometimes get a bit different delays, and I'm not sure why. I mean, you'll see that in the experiment on the screen now, it's also a bit more than 100 milliseconds.
Interesting. Josh Solomon is
I saw his
okay.
face.
So yeah, Josh.
Yeah, Ellie, if you want to leave this for the end, just say so. In fact, that's probably better, but I'm going to forget it unless I say you just tried one level of unbiased jitter for both the cursor and the hand. Might there be any leverage in trying other levels of jitter or biased jitter?
I have tried other levels of jitter, but I haven't ever published or looked at. So when we did pilots, we tried different levels, and it didn't seem to make much difference. And so we went with something nice. I'm not sure what you mean by the biased. So you mean not a random walk?
Yeah. So like particularly in the one with the hand, where you were just jittering the image of the hand randomly left or right? What if it was always more right than left? You know, would they adapt?
Yes, I mean, if you would always also drift to the right, people would adapt. Yes, why not? I mean, just like if you have an accelerate,
Thanks.
if targets always accelerate, you also adapt by aiming further ahead. And I'm guessing if you,
Yeah, this is
I mean, I haven't done that very experiment, but I see no reason why you wouldn't adapt to that, irrespective of the jitter, even.
right. I guess my question was like, would they adapt as fast if there was more jitter, that kind of stuff, you know, Bayesian kind of. Anyway, we can leave that for later. I'm going to forget it.
Okay.
Well, we do know that if they view through a prism, then they do adapt,
Of course.
but then they do adapt to the adaptation, and after a little time, they stop. They figure out the error that the prism is causing. So
Thanks, Ellie.
Yes, I don't know. Did you want to comment on this, or was it just a comment? Because yes.
Yeah,
this is
just forget about it and forward ahead. Sorry.
Okay, I'll go on and we can get back to this later if you want. So then I'm going to start on a different task. So here the task is, it's again in front of the big screen, and the task for the participant is to tap on as many targets as possible within 1.5 minutes. And as soon as as you tap, a new target appears. So it's quite a hectic experiment. That's why it sounds like 1.5 minutes is very short, but it's actually, in some cases, we reduce the time because it's very hectic. Now, what I was interested here is how people would make choices in this task. So we're rushing them, and occasionally they get a choice. So I'll just take you through the 7 conditions, and then you can best look at the left panel and at the pictures at the bottom. So the 2 called no change, which are a large target or a small target, and the large target is also nearby and the small one is far. And what I mean by near and far is, as soon as a participant taps on the screen, a new target appears and it's at a fixed distance from where they tapped, either one of 2 fixed distances, either near or far. And if it's near, the target is big, and if it's far, the target is small. And not surprisingly, it takes people longer to hit the small targets far away than the big targets nearby. And that's what the 2 black dots in A show. So the vertical axis shows the time it takes for each hit. And you can see that for the smaller target, the second one, it takes more time. And the reason to plot this as time taken per hit, when the task for the participants was hit as many targets as possible, so we could have taken number of hit targets, is that all the conditions are randomly interleaved, so the number doesn't tell you much. Then we have 3 targets called replacement, where the target, the first, the more difficult target appears, so the far small one, but then after a brief interval, it's replaced by a bigger one that is nearer. And we did that in a couple of steps because we were worried that maybe a big step would be different than a small step, which is maybe going a bit into the point that Josh was just asking. But it turns out that it doesn't really make much difference. So you can see the open black or open gray symbols show that it takes a bit longer than if they hadn't had to change, because adjusting your movement takes time. But there's not much difference between the different conditions, and that will be even more important when we look at the response. And so then comes the important condition, which is shown here in with purple symbols, the choice condition, which is identical to the replacement that I just talked about. But now when the new target appears, the old one doesn't disappear. And participants are told in advance that there may be at certain moments 2 targets on the screen, and it doesn't matter which one they tap. And in fact, they tap the new target, so the bigger, nearer one, a bit more often than the other ones, but it's about half. It's just over half the number. And as you can see by the purple symbol, so the open symbol represents moving to the new target, and the solid symbol sticking to the old target. You can see that they were actually faster when they moved to the new target. So you might wonder, well, why didn't they always move to the new target? And I'm going to explain that in a minute. I'm just going to tell you the last condition, which is that you start off with the easy target and then the difficult target appears. The reason to include that was just to check that people don't always just move to the last target that appeared, and they didn't. And I think there were in total, in the whole experiment, there were two cases in which the participant tapped on the difficult target when the easy one was there first. I have no idea why they did that. And you see it had no extra cost in terms of time. So the light blue point is at the same level as the leftmost point. Okay, so why do people not always choose the new target? And the middle panel, so panel B, is my attempt to explain this. Now, I told you, of course, that the movements were all over the place, but I've just sort of moved them around, so sort of replotted it so that 0, the yellow dot, is the position of the previous tap, and the black dot called original target is the original target. So horizontal to the right is the progress towards the original target, irrespective of which direction that is on the screen. And then positive values upwards is in the direction of the new target. So the new target could be either clockwise or counterclockwise, but we consider positive in the direction of where that the new target is. And here I've plotted separate in purple now, separately for each participant, where their finger was at the moment that the new target appeared, when they chose the new target in the open symbols, and when they didn't, when they kept moving to the original target in the solid symbols. And you can see directly that there's a separation between these two groups, and so which means that participants considered where their hand was in choosing which target they should move to. So if they had already moved quite far and were moving a bit away from the new target, they kept going to the original target. And if they hadn't moved as far and were moving more in the direction of the new target, they went for the new target, which makes sense. A second finding from this study, which is actually what we did this study for originally, because you may have noticed that this study is actually older than the other study I told you about, is we wanted to see how long it took people to make the choice to decide between these targets. There's a sort of classical choice reaction time thing. And so we determined the response in the same way as I've explained to you before, how we determined responses. And that's also why the new target was not just on the same line to the old target, but was slightly to one side, because then we can look at the movement away from orthogonal to the direction to the original target as a measure of response. And in the rightmost panel, you can see the response for the 3 replacement targets in gray and this choice target in purple. And you can see that the response is, well, practically identical, same latency. So it seems to not cost any time to make this choice. of course, you should realize that this choice, in this case, you don't have to, normally in choice reaction time tasks, that there is a correct choice. Here, there is no correct choice. It's just whichever you do. And it's actually quite good that in our guiding our movements, we can make these sort of choices. Because if I'm going to pick up a cup and suddenly notice that I have to grasp it differently, or if I'm walking and going to step somewhere and suddenly see the dog shit there or whatever, I want to quickly choose a different path. You might think, yeah, well, Ellie, this is really simple. Your choice, if a new big target appears, take it. I mean, on some trials, choose between two things, easy. So we also have a similar experiment where you have to choose between more targets, and that's the last thing I'm going to bother you with. So more than two targets. In this case, it's the same task again. Tap on as many targets as you can. Now there either is one target on the screen or there are 8. And the one target could be one of 5 different sizes. And the 8 targets are so always at the same distance from where your finger tapped again. The different sizes now also always at the same distance, and there could be 8 identical targets around your finger, or one of them could be different. So you can see in panel B in the inset, you can see what it looks like when there are 8 targets. Of course, if there are 1 target, there's only 1 such target. And the participant can tap on whichever they like. It's just the number of tapped targets that counts. And the larger target is shown in this schematic on the right, but it could be in any direction. And the others are then in equal distances from relative to that on a circle around where you tapped. And so now we can look at how participants responded. And so if there is a single target, so that's the gray symbols, then the time taken per hit is smaller if the target is larger. So to the right is larger target. By a larger, I don't think anyone is surprised by this. When you have 8 identical targets, which is the black dot, you are faster than if you have a single target of that size. And this initially surprised us a bit, but actually it's not surprising at all if you consider how the experiment actually happens. So you're tapping like mad on the screen. You can notice that it takes about 500 milliseconds per hit. So you're tapping at a bit faster sometimes than two taps per second. So it's crazy fast. And what you do is you don't tap, wait, see where to go, and then go. You tap and more or less rebound in some direction. And if there happens to be a target in that direction, then you're lucky and you can move straight there. And if not, you may have to adjust the way you move. And that's probably why you're faster when there are 8 identical targets. And it'll become clearer in a minute. If there were 8 targets and one was larger, then again, you have a choice. You may have chosen the larger target or you may not have. And if you choose the larger target, it takes you as much. If you do not choose the larger target, it takes you as long as if there were just 8 identical targets. But if you did choose the larger target, then you're faster. So you're even faster than if there's a single target, and you're faster than just 8 identical targets. And the reason for this is what I just sort of revealed, that you're going to not at random choose whether to go to the large target or not. And you can see that on the right panel. This one shows the probability of a target being chosen relative to chance. And the targets are the angles from what I call here special target, because in the actual experiment they have different special, but special is just large in this case. So 0 means the angle from the large target is 0. That means it is the large target. And you can see that, and 1, of course, is just chance performance. So the larger the target, if you look at the open symbols, the larger the target, the more chance there is that you've chosen the large target. But if you look at the 45 degrees angle, so the next closest targets, you can see that you specifically do not choose those. And in fact, the larger the target, the less likely you are to choose that. And at 90 degrees angle, you see the same sort of thing. But by the time you reach 180 degrees, you're back at just chance level of selecting that target. And so I think this is very much supports the interpretation I just gave before, which is that if you're moving in a direction that you tap, you start moving. If you're moving in a direction that you can still quickly adjust and go for the big target and therefore speed up, you do that. But if not, then you just keep going to the slow target. So this means that in your choices, you're also considering where your finger is at that moment, how it's moving, what all the benefits are, and so on. And this is continuously during the movement, presumably. Of course, I can't. Here, I've only done it at the very beginning, or in the previous example, slightly after the very beginning, but I'm assuming that it will happen at other times as well. Okay, so that's my story. And my conclusion, well, is obvious. Continuous guidance is essential for goal-directed movements. Thank you.
Eli, I want to thank you very much for a really interesting talk, and I want to invite everybody to, first of all, unmute yourself, and let's give Eli a big applaud. And I myself have questions. I see Josh too. So if you're happy, is it okay to open the floor for questions? And
of course.
Okay, lovely. So
should I stop sharing or should I stay sharing? And
I don't know, whatever you prefer. Maybe let's see what Josh is asking and if he wants to refer to the slides or not.
So, thanks, Sharon. Just a question about the when there's just one target in that last experiment, Ellie, could that be a completely ballistic? Like the reason it takes longer is people compute where they're going to move and then they move with no correction when there's just one target?
Why would they do that?
Well, so there are two things, right? So they're trying to hit as many targets as possible, but they're also trying to actually hit the target and not miss.
Yeah. Yes.
So if there's just one target, maybe, I don't know. We have to come up with a reason for why it takes longer when there's just one target.
Well, I think it just takes longer because you happen to be moving in the wrong direction on some cases. When there are 8 targets in all directions, it's very unlikely that you happen to be moving in the wrong direction because there's always one somewhere you're moving.
Absolutely.
I'm personally not a believer in ballistic movements at all. I think for a movement to be ballistic, it has to take less than 100 milliseconds. Because
it may take some time to compute where to launch. But so I guess so that's something that if we could think of a way of demonstrating that's not, you demonstrated it's not ballistic when it's jittering, right? That's solid. But here it's not jittering, all the noise is internal. It just might be different. Anyway, that I'm just
so that's why. So we're getting back to the point you made earlier. So that's actually why I think it's important to also show this for the moving targets where you don't, where as a person, at least you're not aware that it's jittering.
Yes. Good point. Yeah.
So it's not, but none of these things can be conscious. I mean, in this experiment, it's really crazy because you're tapping like mad, and you sometimes realize, oh, I didn't go for the big one and went to the one next to it. But you think of that, you're already 2 taps further, because thinking about things takes a huge amount of time compared to tapping.
Okay. Thanks. I'll
Yeah.
lower my hand. Let someone else go.
Gabriel?
Gabe?
Hey, Ellie, great talk. Thanks. So I got a question for you about just actually, it kind of really piggybacks on Joshua's comment. So the observation you offered is that when people are moving in the direction of a new target, they're slightly faster to get there. And I wonder if you consider that to be purely like biomechanical savings that reduce the time, or is there something a little more complicated going on? Something about, I don't know, eye movements moving in the direction of the target, or just something about kind of already having anticipated their, predicted the way their hand trajectory will evolve while continuing along that current trajectory, and have you considered investigating that?
I think it's biomechanical, as you said, but I wouldn't know how to investigate that. It's there's always been going to be a correlation.
I agree, and that's why I asked the question. See if you had an idea where I was drawing.
I would have to think about this. I'm not. I would guess biomechanical. I feel that it's just your arm are moving in that direction. Of course, there are some subtle other problems which I didn't talk about. So sometimes you don't move to certain targets because they're hidden by your hand. That's why my time taken per hit is actually a much more complicated measure than you might think, because it's the median hit time corrected for the number of misses. Because if your hand is in front of a target that appears, you're obviously not going to move to that target. So your arm sometimes covers it, and then you move away, you see it, you adjust. So there are some other things, but I would think that the main thing is biomechanical.
Okay, thank you.
Lovely. I also wanted to ask, actually, you just referred to that, but because if I'm not mistaken, you didn't show us the analysis about the misses or the errors that people make. And I mean, because if you have so many of them, you probably are not very precise. And I wonder if that has to do, I mean, if there's any relationship between the errors you make and, I mean, I'm talking about the multiple targets appearing at once. Whether spatial location in the the spatial location relative to the center would interact with the error spread or the error size. And I assume that there are like more misses or going to the bottom targets because of the hand. So I'm just wondering if there's anything about that. Would be
No. So we've looked at effects of position on the screen. So the main difference you see is when when you need to go to a target that is for right-handed people at the right bottom. It usually takes more time because your hand is in you see it later. I mean, your hand is in the way. You sort of you have to curve around so you can see it. The misses, there aren't all that many misses. There are misses. There should be misses. We've shown this before, that the optimal way to move is actually to miss quite a few targets, because you need to find a balance between slowing down to hit targets and moving fast. And so that's why so this data all includes misses. So it's the time taken per hit, which means that we do add the time of misses to the time taken. but
And
then divide that time by the number of hits.
I see. And do people make corrective movements? Let's say, I want to hit one dot, but then I land. Well, not I land. The finger is
Yes.
just
So initially, so we give people a bit of practice, and in the practice they try, but they can't, because as soon as they tap, a new target appears. So initially, participants try to correct their movement and go to where there is nothing, and then the target jumps again. But that once you've seen that doesn't work, they don't do it anymore.
I have another idea. Not that I'm just thinking, do they use their binocular vision to do this task? I mean, I know it's a plane, like it's a surface. It's not something that is changes in depth, but still, it's in their peripersonal space. And I wonder if people would be slower, or they would catch up really quickly in a learning curve if they would have to use just one eye. Just a thought.
Yeah, it's a nice thought. I have no idea. I don't see why it would be fundamentally different,
Yeah,
but
maybe not.
you can. I think in the other experiment, the not crazy tapping experiment, there's a little bit more chance that it would matter, because when you tap, you have to time your tap, so you need to judge the distance to the screen.
Mhm.
And although you're standing in front of it and it's always at the same place and you feel it each time, you might be a bit better in judging that position when you look with two eyes. So it might be an advantage to have binocular vision. But I don't know. I've never tried. I've tried with and without binocular vision in interception tasks when a ball is moving towards you. Yeah, then it helps. But I've never tried it for this task.
Interesting. Thank you. Are you still okay with taking another question from
Yeah, sure,
Kelly?
Ellie.
Yeah. So, Ellie, you need to unmute yourself.
Okay. Hi, Ellie. This is the other Ellie with a lot
Yeah.
less hair. And at the beginning, right away, you said that this is going to be a lab experiment and not in the wild experiment. And
Yeah.
I'm going to try to drag you back in that direction, because that's where my interests are. And I there are probably many situations where people aim at colliding with a target or meeting a target, but I'm interested in avoiding collision. And even your one example that you gave about the dog shit on the included avoiding it rather than colliding with, which may be quite different. And so I'm asking if we can learn from all this anything about the way one would avoid the collision is the desirable action, rather than, you're okay.
Yeah. Okay. So I'm still not going to go out of the lab.
Yes, yeah.
But we did an experiment to see whether people would actually consider obstacles when they do these responses, and actually whether they would consider the obstacles positions at the very moment of the response. So this is it's a complicated experiment, but I'm going to try and take you through it quickly anyway, because you already know what responses are and all that. So this is again you move to intercept a target. In this case, again, slide across the screen because they're virtual obstacles. So you start at the green dot, and then the black dot is the target. It's moving across the screen, and your task is to intercept it when it's in the gray region. So you have to move between these two obstacles. And then at a certain moment, the target gives a small jump. I mean, here it's quite a big jump, so you can probably see it. And at the same moment that the target jumps, the obstacles either come closer together or move further apart. And so our reasoning was if you actually are considering the obstacles within this response, then you should respond more vigorously if the obstacles move apart than if they move closer together. And that's indeed what you see if you look at the right side. So this is just the same this is the response to the target jump. And the green one is for when the obstacles move further apart. And the yellowish one is for when they move closer together. And you can see that they're different, that you respond more vigorously when they move apart. And you can actually see that in all participants if you look at the inset, which just shows the average of the gray bar time period. So yes, I think obstacles would be exactly the same as moving towards targets. It's just I study mainly moving towards targets.
Okay, thanks.
Last, Josh, if that's okay, just last question, and we'll sum up.
Back to moving towards targets. Would you think there'd be any reason that saccades would be qualitatively different, Ellie, or just more of the same?
What?
Instead of intercepting with your finger, intercepting with your fixation, like a saccade.
I would expect it to be well. I mean, saccades are ballistic in the sense that they are too short to adjust while ongoing. I mean, you can probably adjust them by presenting something before, but so I'm not sure in which way you're asking me whether it would be the same. It's
yeah, I'm not sure either. That's okay. Okay, thanks.
Eli, I want to thank you again for joining us and for a really interesting talk. So thanks again. And I want to say that next week we're going to have an in-house talk by Professor Pina from Italy. I hope we'll be able to broadcast it. It's going to be about visual illusions that he has created. So I will keep you posted if we're successful in broadcasting it on Zoom as well as in-house. So thanks again a lot, and have everybody have a great winter holidays if you're not here next week.
okay. Thank
Lovely.
you.
Thank
Goodbye.
you. Thank you very much. Bye-bye.
Bye.